Good evening, every College of San Mateo. Thank you so much for joining us on our second speaker series of the semester. We have two terrific presenters that will be sharing their path through STEM research and career. Just a quick reminder is that you could access our previous presentations by going to our webpage, which will be posted on the chat. The recordings will be there, and you could also go to that page to join us for our next event, which will be in a couple of weeks on November 3rd, the same time. Now, without further ado, I would like to pass it on to Panos, our moderator, who is also a Stanford graduate student studying immunology. So take it away, Panos.
Thanks for the introduction, Olivia. My name is Panos. I use he, him pronouns, and I'll be moderating today's panel. I'd really like to thank Olivia and the MESA program for all their help in putting together this speaker series. I'd also really like to thank Cecilia and Vali for serving as our second pair of panelists of the year. So today's panel will start with Cecilia and Vali each spending about 10 to 15 minutes talking about their career paths and their current work, and then we'll transition to about 30 minutes of Q&A. So please feel free to submit questions at any time by clicking on the Q&A button at the bottom of your screen, and then I'll read them out loud at the end for Cecilia and Vali to answer. So now we'll jump right in, starting with Cecilia. So I'll hand it off to her.
Thank you, Panos. Let me see if I can share. Oh my, it was working before. Sorry. Yes, there we go. Can you see the screen?
Yes.
Okay. So good afternoon, everybody. My name, as Panos introduced me, is Cecilia Ramello. I am currently working as a postdoctoral fellow in Crystal Maker's Lab in Stanford Cancer Institute. And I'm going to tell you a little bit about my story, so you can, Panos, tell me if I'm running low on time. I grew up in Argentina. For those who don't know, Argentina is very south. If you go till the end of the American continent, we actually have the city and the train of the end of the world. But I grew up a little bit further from here in the Mediterranean province of Cordoba, in a very, very little town that is called Elena. According to the last census, Elena has less than 3,000 people living there. Most of my family and my friends from my childhood are still living there. But to put you in context, Elena, which is in the countryside of my state, is smaller than a Stanford campus, and it has barely the same amount of postdocs that Stanford has. So unbelievable, right? As you can guess, the diversity, the intellectual challenges, and the opportunities that I grew up with were not many. And the high school, the only high school that my town has, it was the core of that high school was social sciences, while I was enjoying more mathematics and chemistry at that time. So 2003 was the year where I finished my high school, and it was a pretty rough year for me because I had to decide my future back then. And it was rough, and I was very lost, not only because the high school had given me the tools to feel familiar in a field that I didn't enjoy much, but also because I was the oldest of my siblings, and I was the first one of my family to go to college. My mom and my dad, they did finish high school, but my grandparents, for example, they just went to elementary school, and some of them, they couldn't even finish. So if I see my family like in a bigger picture, I had some cousins that were studying at the moment where I was ready to go to college, but 3 of them were studying psychology. One was business administration and another one was veterinary. So I was really missing those professional role models to help me dive into my skills and potential careers. So I take some aptitude, career aptitude tests, and they were a total failure. So what I did was a list for myself of the things I was doing during my high school and I was enjoying. And some of those things were that I was participating in the Olympics of mathematics, either alone or in a team with other friends, and I like it. I was giving some support classes for students in different areas of the school, just helping them even with English, believe it or not. And I was also volunteering in my town's hospice for senior people, and I was just there just reading, painting, or sometimes playing cards, for example. So by using that list, I just, and looking up at the classic career path that I could select in Argentina, I had this short list, and I crossed out immediately engineering because honestly, I didn't know anybody who was an engineer at that time. And talking about the STEM careers was not something that I heard about at the moment. So those were the only options I had. So I decided to do my first big research in my life, and I went and I interviewed the MDs, the 2 MDs that we had in my town, the biochemist that happened to be my chemistry professor as well, and my mathematics and my biology professor, and to discuss about their experiences. Fun fact, most of them were men, and I'm still remembering my conversation with the MDs where they say that choosing that career and to actually be a good doctor, you have to live for that career. And they say that that wasn't very compatible with a mom or an spouse. That was terrible to hear, honestly. But eventually, at that moment, I didn't know anybody who was a woman and a doctor. So it was kind of a big piece of advice for me. Bad, wrong, I know. But that moment, that was what I had. So I decided to go for a bachelor in clinical biochemistry, but always thinking that if I didn't like it, I couldn't just drop it and move to med school. And lucky for me, I really liked it. And eventually I learned that I liked more biology than chemistry later on. And when I took my first immunology subject, I found my really passion. What a clinical biochemist does in Argentina is to work in a lab that is going to help with the diagnosis or the follow-up of a treatment in a patient. So before getting my bachelor degree, I worked for 1.5 years in a hospital. And what I was doing there was just collecting blood from the patients, and once in the lab, analyzing that blood or other human samples like urine, stool, spit, whatever you imagine. And the analysis of those samples was consisting in taking a look at those samples under the microscope or quantifying analytes, for example, glucose or some liver enzymes by using chemical reactions. And I really enjoyed that work, especially the work of looking the samples under the microscope, because I was fascinated with the fact that even with a tiny drop of blood, after a staining with a mixture of dyes that we were using, we were able to see this in the microscope and identify a normal blood compared to other diseases where you get abnormalities. Or we were also able to see if someone had an urine infection or probably a kidney malfunction, just looking at the urine under the microscope. I enjoyed a lot. I was fascinated, but the lab work was eventually very routine, and I felt like I needed more. So in that more, eventually I discovered that was science. So I decided to pursue my career in science. And following my passion for immunology and my question, because I wasn't able to understand why having T cells in the body that are able to fight infections and succeed many, many times, in most of the cases, when those T cells are trying to fight against cancer cells, they fail. And eventually, I learned that the tumor cells can use a variety of different mechanisms to avoid those T cells to kill tumors, right? So I was fascinated with that, and I wanted to study this field. And I was very lucky that during my search, I met this woman, Carolina Montes, who was coming back from her postdoctoral work here in the US and opening her lab in Argentina, in the university where I did my bachelor degree. And she was part of this bigger group led by Adriana Gruppi and Eva Costa, all immunologists, and this amazing group of people that joined the lab right after me, that became my scientific family, I believe, for life. And the work that Carolina was doing was kind of studying these evasion mechanisms that the tumors can use to avoid T cell responses. And she was describing how tumors can induce T cells to become older, or what we call senescent. And so I decided to join her lab. But before that, to start my path in research, I applied in 2009, just a couple of months after I received my bachelor degree, for an immunology teaching assistant position in the university. And this allowed me to go to the research lab and volunteer. And that year, while I was teaching and learning research and writing my fellowships, I finally got admitted as a PhD candidate in 2010 under the supervision of Carolina. By March 2015, I was defending my thesis. I was pretty happy because at the project level, at the moment, I had given the senescent T cells an identity and described how they function, and had my first publication. At a scientific level, I learned a lot of techniques, a lot about basic immunology and how to apply the scientific method. But eventually, the most important thing that without a team, there is no success in this STEM world. And at a professional level, I improved my public speaking skills, and I got a sense of self-confidence that I didn't have before. And at a personal level, I was able to experience the importance of the public education, the impact that peer mentoring has in to stay in this academic environment. And I also experienced profound changes in my personality that was really positive. So my career was looking like this. Some pieces were fitting, but the still work in process. And I was feeling like I wanted to continue my career in academia, but Argentina was just not enough for me. So I wanted to go and work in a foreign lab, but at that time, my husband was still finishing his studies, so I had to stay in Argentina a little longer. And in the meantime, I applied for a fellowship to work with human samples. So with this fellowship, I generated like a bridge between my research lab and two hospitals, and a local hospital in Cordoba and a hospital in Paris, where I got to work with samples from breast cancer patients that didn't receive any treatment, but the first line of treatment was surgery to remove the tumors. So I got to collect peripheral blood and tumors and other samples from those patients and compare them. And eventually this experience taught me a lot about ethics and when working with human samples, how to discuss science with a more interdisciplinary group. And at the end, my career was looking a little bit better, but still some missing pieces. And what I wanted to learn at that moment was tools or techniques to be able to manipulate those T cells that I've been studied for years. So I decided to join a lab here in the US at Moffitt Cancer Center and Research Institute in Florida, and the lab of Daniela Batedaga, to work in CAR T cell therapy. And this was an amazing opportunity for me because this therapy was pretty cool, but as well because I would learn how to manipulate genetically T cells. So the way that CAR T cell therapy works is by using the patient's own T cells that are isolated, and then within the lab, we reprogram these cells by adding to those T cells genes that are going to encode for these chimeric antigen receptors. The chimeric antigen receptors are proteins that don't exist naturally, but that we generate in the lab to comfort the T cells with the ability to recognize tumors. So once we reprogram these cells, we put them in a condition that allow the cells to grow in high numbers, and then those cells go back into the patients, and we expect them to kill the tumors. So at Moffitt, what I was doing was trying to understand how these proteins that we designed in the lab were transmitting those signals and were telling the T cells what to do once they spot a tumor cell. And it was a really nice experience because I acquired a complete new set of skills. I learned all the molecular biology techniques that I know. I learned how to genetically manipulate the T cells that I was looking for, but also I learned about proteomics, about mice models of cancer, and I had the great opportunity to mentor undergraduates and graduate students, and it was an amazing opportunity. But after 3 years, I came to the Bay Area and I joined the lab of Crystal Maker's lab in Stanford, where I continue my work in the CAR T cell field. And you already saw this picture. These were the mechanisms that tumor cells use to avoid T cell responses. And CAR T cells are cool, are pretty cool because they can avoid or bypass some of those mechanisms, but some other mechanisms that tumor cells use to dampen these responses are still there. So what I'm doing in the Maker lab is to investigate new possible strategies to diminish those negative effects of the tumor cells on these CAR T cells. And the truth is that Stanford was the first place, or was the place in the top of my list when I was deciding where to go for a postdoc before leaving Argentina. But at that moment, my husband and I it was not possible for us to afford moving to this place. So we decided to take another huge opportunity and go to Florida. And once ready, we came here. We finally did it. We are where we wanted to be. And I think that what helped me a lot along my career to be where I want to be is to always be critical of myself first, and also my experiments, to accept that the experiments can fail, but that doesn't mean that I am failing as a scientist, to learn to work with others, different backgrounds, with different interests, always ask people for help, particularly when I am taking a big decision. I learned to accept feedback, and particularly positive feedback. That's harder for me than the negative one. And also, everything I've achieved is because I have a network of people that I trust with my heart that is there to support me. And of course, having my heart in the work that I do, because I love it. And that's it. I hope you guys have the best of your luck choosing whatever the path you choose. And this is my mail and my LinkedIn. I'll be happy to receive any story from you and help in any way. Okay. Thanks.
Thanks so much, Cecilia, for that great presentation. As a reminder, you can submit questions at any time in the Q&A. We'll ask them at the end, but for now, I'll ask Vali to go ahead with his. Thanks.
Thank you, Panos. Cecilia, that was amazing. I find it so cool how diverse of backgrounds we have in the lab and also in this field, both culturally, scientifically, intellectually, as you can tell, perhaps the personalities and the passions that drive us to pursue careers in science, technology, engineering, and maths, but also in careers that end up helping people directly. So I'll show you a bit about my story in sort of a similar fashion. I was born in Romania in the 80s, and my parents wanted to flee communism, having lived through that in our family's history. They had previously come over to Ohio, but moved back. And in the setting of social upheaval and less opportunities, they thought, how do we give our family an opportunity? And so pretty quickly they were able to come over to Cupertino in the early 90s, where my parents, having had some degree of an engineering background in computer science and electrical engineering, came to Silicon Valley, and I grew up here. I attended undergraduate school in UC San Diego. I focused on bioengineering. And shortly after undergrad, I joined a company called Illumina that does what's called next generation sequencing. And that is a tool that helps us measure the base pairs of our genome. And you can imagine that's useful for understanding a whole genome of a person who has their genome sequenced for a diagnostic application or for a mapping application. Or as you'll see in the couple of examples I'll show you in my own career, sequencing can be useful as a tool for counting. It can look at mutations and trend their abundance or their emergence or other features in which sequencing doesn't just measure the genome, but measures it in a specific clinical context. So at Illumina, my task was to optimize workflows that help the throughput of some of the core chemistries that enabled sequencing. And in this example, you can see here, what I did was look at input variables of time, temperature, extraction, mixing, as well as different chemistries. And what I plotted were surface response curves through what's called a DOE, or a design of experiments, to determine what was the optimal combination in an engineering approach to make these sequencing reagents more reproducible, cheaper, and then from a company standpoint, obviously more profitable and sustainable to enable further R&D and launch novel products. But from that experience, I wanted to know really, how is this going to be translated into patient care or impact people? So I picked Illumina's biggest customer for medical school. And at that time, it was Baylor College of Medicine, who was running whole genomes and whole exomes in clinical settings. And during medical school, I crossed the street over to MD Anderson Cancer Center in this area of Texas Medical Center, and was exposed to the ways in which molecular tools were being applied in patient care in one of the most premier cancer centers in the world, where they have access to these tools, these skill sets of how to interpret this data, and then obviously the most in need patients who have gone beyond the standard of care for treating their cancers. In that group, I had my first exposure to really what clinical research meant, and I worked on developing a plasticity signature that explained the aggression of cancer using some of these tools. I then proceeded to residency, which is the next phase of training after medical school. I picked pediatrics for, I guess, a selfish reason. I enjoy my patients. They're absolutely wonderful. They keep me humble, and I love going to work. It's called a job, but I feel like it shouldn't, I don't feel that way. I feel like I really enjoy going to work every day. I also viewed pediatrics as a way in which the fields of germline sequencing and cancer sequencing would converge, because increasingly we're going to be able to anticipate which mutations or variants predispose people to have cancer, or how we really detect that cancer before it becomes present. So I thought to myself, to be at that inflection point, I'd like to be working with the younger population so that one day we can use these tools to detect and prevent cancer. After residency, I then attended Stanford for fellowship in oncology. Pediatric oncology specifically, and last year finished that role. And so what I've learned over these years, to get right to the point of the lessons, was that finding a vocation or your way through a career, for me, has really been an overlap of being very honest with what my talents are, who my mentors are, and those have evolved over time, and then really honing in a work ethic of doing this day in, day out with a passion and with a grit that when it gets hard, or when you fail, which you will often, you can get back up and develop that skill set. So really what I wanted to learn and reflect in this presentation was, what did I learn from the instances where there wasn't this overlap? Because we can see this Venn diagram of where talent and work ethic and mentors all come together. But what happens when they don't? And in my experience, as you draw that arrow to the right, I experienced failure in every single one of those words there, whether it was not reading enough of the literature to know things in the clinic that I should have, not performing well in labs or shelf exams, having a subpar GPA, not impressing people in interviews, taking the MCAT 3 times to ensure I got enough of a score to justify my academic credentials, experiencing industry as a path to medical school prior to going directly and feeling like that was actually additive to my career and professional development. Obviously taking innumerable exams in the form of shelves and applying for grants and board exams, etc. But at the output of that, I can say, focusing on developing talent in being a good student and applying myself every day, and then seeking mentors in all of these axes where I couldn't get these overlaps to fit in, has enabled me to progress in my career and become a physician. And one that applies the technology I helped work on early. So just to go pretty quickly through a couple of the examples, we use next-gen sequencing now in the clinic. This is an example of one of my patients' next-gen sequencing trends to very, very sensitively detect the presence of disease. You can see on the left, the burden of leukemia was very high at diagnosis, and we were able to eliminate it using standard chemotherapy, followed by CAR T-cell therapy, which attracted me, just like Cecilia mentioned, because of the ability to escape the toxicities of chemotherapy and perhaps offer something much more targeted to the cancer. And what we're reassured here is that when we're trying a novel therapy, we can continue to prove that the therapy we're trying leaves the disease in a remission state or undetectable. And we see these are called Kaplan-Meier curves, and they show the probability of disease-free survival or overall survival on the right, and the separation of curves of patients who remain circulating tumor DNA negative and therefore are more likely to survive as opposed to those who are positive. And this has been shown in innumerable clinical trials. And so what really motivated me was to develop an assay in which we can take this paradigm of precision, personalized molecular monitoring that we know correlates with outcomes and bring that into clinical trials where we're trying new therapies using the engineering approach that I'm somewhat biased to want to apply in my career. And so my first role as a fellow was to track the mutations of the specific mutation of a brain tumor in the patients receiving CAR T-cell therapy for diffuse midline glioma. That mutation you can see when you're looking at the normal versus the tumor cells appears in where the arrow points, and that is what's called an H3K27M mutation. What I helped develop was a technology that, again, went to industry to look for the tool. This company, specifically Bio-Rad in this instance, developed a tool called digital droplet PCR. And what it enables us to do is separate the detection of that mutation in the tumor versus the normal. And so what we can do is design primers using literally sequencing right here of that genome and identify the wild type versus mutant presence of every single read in the cerebrospinal fluid of patients receiving this therapy over time. What I learned in developing this assay was, number one, you have to be very rigorous. Like I was showing you the validation and the experiments I did at Illumina previously, I applied in doing the same here to demonstrate that the measurement of this mutation over time is reproducible. What you see in this curve right here in the blue is the linearity with which we can measure the mutations over serial dilutions over time. The R squared of 0.99 essentially says that at any one point, I can quantitatively and reproducibly show you how many mutations are present in a sample using this visualized workflow here of digital droplet PCR. Of course, this is team science, and I pointed out earlier when mentors happen. So with the help of Elena Sotillo and Sneha Ramakrishnan in the lab and under the mentorship of Crystal Makel, I was able to demonstrate that we have a very high sensitivity for measuring this mutation in both negative and positive controls. And with assay optimizations, and I'm sharing these slides just to show you the degree of work that goes behind one very simple curve, which is really what was the mutation trend in these patients over time. And in this left figure here, you can see for different patients, whether they received in gray an IV infusion of CAR T-cell therapy or in green an ICV or intracranial ventricular infusion, the kinetics of the mutation over time And what's encouraging is you see after an infusion, commonly after an arrow, you'll see a spike in the number of mutations you can track in the cell-free space. And what we think that demonstrates is the on-target killing of the tumor in ways we otherwise wouldn't have been able to measure as specifically to the disease. The alternative being, of course, imaging by MRI. The other quick project I'll just take you through 5 more minutes is the ability to apply the same tool to measure mutations in leukemia and as well as simultaneously detect microbial species or infectious complications or causes of leukemia in patients. Again, this was in the context of mentorship on the right here, Chuck Gawad, David Klein, Yun Tao. In this group, I helped develop an assay where we looked at the concordance or the shared ability of detecting these mutations in the bone marrow where leukemia originates, in the peripheral blood where it's much easier to obtain serial samples, or in the cellular space of the bone marrow that represents the true residual leukemia cells. Like the other previous assays I've shown you, this required much validation. We demonstrated that there was uniform and deep coverage throughout the entirety of the chromosome. You can see here chromosome 1 to chromosome 23, as well as the yield of cell-free DNA over time, which qualifies our denominator, how much DNA is going into an assay, so that we know we have the sensitivity to claim that if we're measuring, we're going at a depth to catch these rare events of mutations over time. We describe our patient cohort, and you can see over the 20 cases that we sequenced, the number of variants by deeper and deeper blue color over here, and the variation in the variant allele frequency, or the rise and fall of mutations over time, again demonstrating an ability to measure and capture the temporal biology of the leukemia in response to therapy. We also showed that across the peripheral blood, the non-cellular bone marrow, and the cellular bone marrow, there was variable ways in which you could capture this information with high fidelity. But interestingly, we developed an additional variation on this using the same tool of counting to design baits that would literally hybridize or catch regions of the genome that are circulating in our blood right now that correspond with microbial species. And what we demonstrated using this assay was the ability to detect in patients over time, over multiple time points, the rise and fall of microbial species, whether they were viruses, funguses, or bacteria, all of them being DNA species, that correlated with the degree of immunosuppression that they had because of chemotherapy. We again demonstrated that different species could be profiled, again, reproducibly over time, and that different compartments had varying levels of these species, all using the same tool of sequencing applied on different sample sets, but over time in patients receiving chemotherapy. And we validated the different regions that would be most sensitive to obtain this information in the future. Because again, the alternative for these patients is to stick a needle in the bone marrow to obtain this information. We demonstrated that in many instances, next-gen sequencing, because of its sensitivity, could as well detect this information from the blood. So in thinking through these experiences and really my reflections, I think I'm echoing pretty much verbatim what Cecilia mentioned in terms of her core thoughts of how she progressed in her career. My first one is really that picking the right thing to work on is the most important element of productivity and is often ignored. I feel like we commonly are willing to just go without having thought, what am I doing? What do I want out of this experience? Who am I doing it with? And am I working on something that after I spend some time on it, I can reflect honestly and think, okay, did I get something out of it? Either personally or contribute to the field or whatever your motivation is for, why you're in school currently or what you want next. The next part is none of what I did was done in isolation. I needed mentors. I needed my friends, my family, my spouse. And I learned in this process, specifically in science, that team science is fun. Also really productive because you're not doing it alone and you have the mentors that can support you in learning what you don't know. And the further you get along in your career, you realize how much you never knew. So in my clinical career, the combination of clinical and research mentors was key because each have their own strengths and perspectives on how to get there. So my driving mentality was just try to be around smart, productive, happy, and positive people that don't tell you can't do something. And in my experience, I'm showing you all the highlights of things I've accomplished, but I can promise you there were 10 failures for every one positive thing I've shown you succinctly. A pandemic lesson, which I think is probably most near to us in the past year or two, is really to modularize projects and prioritize in a way that generates momentum. By that, I mean, you can do 2 or 3 things in parallel because of the time you're given, the less commuting, the fact that we had social isolation and distancing, etc. And overcommitting a little bit can be good, but I've also learned that overcommitting a lot can be bad. If timelines slip or you're overstressed or you don't have time for exercise or sleep or hobbies that help you reset. And then lastly, you can really learn anything you want and you can get better quite quickly. I think I knew what CAR T cell therapy meant 4 or 5 years ago, but now I'm infusing those cell therapies in patients in the clinic and understand them to a degree I never would have thought I'd do. And that's primarily from the combination I reflected on earlier of mentors, work ethic, and talents or passion about what really motivates you to get out of bed and to pursue a career or a certain direction. And so what I really thought was an unlikely miracle the first time seems like has become a formula for me. And from all that, I just reflect that the mentors I've had here at Stanford and the experiences I've had of taking care of patients with my co-fellows, the nurses, the attendings, has really been a rewarding career in a pathway that built upon an engineering background, and then with the support of mentors and really a lot of hard work and determination has enabled me to increasingly contribute from having learned over the years how to apply these skill sets in clinical settings.
Thanks so much, Vali. That was awesome. We already have a couple questions for students for you and Cecilia. So we're just going to dive right in. Feel free to submit as we begin. I'll just ask them in the order that they're asked. So the first question is either Cecilia or Vali or both feel free to answer a subject matter question. Have CAR T cells been shown to cause remission? And if so, how effective is the therapy currently?
Sure. So clinically, we use CAR T cell therapies approved by the FDA for leukemia lymphoma. The challenge is being primarily that we are targeting the immune system against something that's very specific to the tumor. And the tumor often looks like normal cells. So the big challenge is, can we find other targets or other ways to make that on target, on tumor effect long-lived? To answer the question quantitatively, I tell my patients who do express that target, CD19 is what it's called for leukemia, that about a third of patients can have a durable or long-lived response to therapy. But there are many, many challenges, and this is why we work together in team science to broaden that therapeutic index to a lot more people and have that effect last a lot longer.
I just can add something else to that. And what we can also see from the CAR T therapies is a relapse. And that can happen because the way the CARs are built, they are going to target an antigen. So if the tumor loses that antigen, then the ability of those cells to kill the tumors is also lost. But it's a very promising therapy.
Great, thank you both. Next up, we have perhaps a difficult question, but if you had to choose, what was your most difficult obstacle and how did you overcome it?
I have an obvious answer, and that's exams. I hate memorizing. I was taught as an engineer to not memorize a thing and to derive. Even though I remember names and people and like experiences, it's very different to memorize pathways in biochemistry and things that you're otherwise as an engineer very willing to go look up or Google, as we all do everything now. I think we Google the time and the weather rather than just go like guess it. So for me, it was exams. The way I overcame it was to study with friends, make flashcards, take practice questions, and fail a lot. I took many of my exams many times, and it was extremely frustrating and demoralizing because I knew the information. So I had to teach myself a willingness to make flashcards, do it again, be committed, and really spend the time working on a weakness, which for me was forced memorization when I told myself I could otherwise look it up.
So I think the most difficult thing I had to do, I don't know, I think was the decision when I came to this country, I came thinking that this was a transition period for me, and just to learn some expertise and then go back to my country, to my family. And eventually, I think the most hard decision for me is to decide to stay here. No looking anymore something in Argentina, and just because it's a difficult decision, but it's because I found that the opportunities here are amazing. So I think that was my most difficult obstacle. Mm-hmm.
Thank you. So the next question says, I want to be a doctor. How did you know that being a doctor is exactly what you wanted to be? What motivated you to go into the medical field? I'm actually going to broaden this a little bit into how do you know which path you want to follow to contribute to medicine? And you don't necessarily have to go to medical school in order to contribute to patient care or to scientific discovery. So if each of you could address that, especially since you both work in the same lab but have different training backgrounds.
Cecilia, you go.
I go? Okay. Yeah. So, okay. I think what I like from this career and from science is just to understand how things are, how biology works, how a cell works. So that's my purpose. And that's what drives me every day to go to work and do my experiments and just be motivated. Yeah. The why.
For me, I think I'm innately curious and I like working with people. I also like deconstructing problems in my engineering way of understanding how it works and what we can do about understanding that. And for me, cancer being something that you could then measure and address and engineer and think about, it's a very rewarding hybrid of the two domains for the way I think about it. And there's many types of physicians out there, but the skill set that I've known I wanted to pull through was this curiosity and specifically about how things work, and with an understanding of how they work, can we affect them, and can we do so with people? So I think it's really a domain plus the ability to work with people in that application that makes my path and commitment to medicine have always been something of obvious to me. And I'd say that from every experience made it more and more clear that that's a path I want to take, that makes, again, going into work not feel like work, feels like I'm doing it because I do it anyway, but don't tell whoever's paying me that.
Thank you. Our next question, I love this one. How did you go about seeking mentors, and what were the characteristics you sought out in them? And what if they were hesitant in assisting you? How did you troubleshoot that to get the assistance you needed? And just an add-on to the question, if you were looking for someone to mentor, what would you like to see from them, and how would you help them along?
Oh, sorry. Okay, I go. So, what to look in a mentor? I could look in a mentor, someone who can really hear what your needs are and be there for you, not to tell you what to do, but to help you find what you're really looking, but you still don't know what is that. And yes, but the question on how to find a mentor, I found that difficult as well. When I was in my country, I found that easier than here. And I think the reason is because here you have different cultures, different ways to approach people. But in my experience, always when I ask, I received a response, whether or not they say they have time or not to help you. So don't be afraid to ask. Don't be afraid to reach out. And whenever you feel that relationship with a mentor is not productive anymore, then you can just say it and find another person.
Yeah, I feel like this question, or maybe it's our society or or training programs always try to formalize mentorship. And I've never found it to be something that needed to be so formalized as it seems to be made, even though I get it and I even put it as a word in my Venn diagram. But for me, the mentors I can think of were people I admired. And initially they were the executives of the sequencing company that I asked, would they get coffee with me because I'm interested in their path. And I just wanted to learn about how they got there. Followed by, in medical school, the doctors that I admired, how they interacted with patients, and on in residency and fellowship, the other colleagues of mine who did things better, caught diagnoses earlier, ordered the right labs, thought about the right differentials. So again, all driven by admiring people whose skill set or approach to things I wanted to learn. And I didn't ever really formalize like, you're my mentor, right? But the other part I can think of is increasing as I'm training, I think of my patients as my mentors. And I say that because they really challenge me in ways to communicate more simply or to get at really what their problems are more directly. And I feel like that's something that I've come to appreciate, that mentors need not be some person that's the most senior in their field and potentially unreachable or so busy that asking of their time is perhaps not as meaningful to where you are in your career than simply who do you admire? How can you interact with them in ways that you learn from them? And in doing so, it's just over time and with relationships that mentorship experience is born out more of your interaction and friendship as opposed to some formal assignment.
Thank you. So our next question is, I know both of you have discussed this. How do you find what you want to research, especially given everything that you can potentially work on and the hyper specialization of research these days? You really have to find that one question that motivates you. So really, how do you find that question that you're going to pursue?
So I think there's so many questions that I often struggle with this as well, and that's probably defining scope in any one day, week, month, year, project, et cetera, for what you want to accomplish. And I really think of how can you measure progress that you're getting to the right place in whatever you're committing yourself to, whether, and I'll answer the research question, but I think it's really, what do I want out of this? And am I being realistic with the time frame and the skill set I personally have, or I could build with a team to do it best? And I really focus on that part in research of doing it best, because if you do it okay, it's not going to really move the field forward. You have to be the best at what you do. And so when I think of defining scope or defining a research question, I again pursue my own curiosity of what I think is a problem with the field. So when I thought about next-gen sequencing applied to patients, I was frustrated, and still am with most relapse cases, that we don't understand what mutations we could target, but just go ahead and treat with chemo and see if it works. That frustrates me. So with that innate need that I have to be able to do things more mechanistically and prove rationally why we're doing it, I propose those questions to people who are in the field and say, well, what if we did this? And they cut me back a decade into reality of what we need today and say, well, why don't we prove that if we do it with a few cases, we have a signal that we could then expand further, or it's realistic to fund or to publish, et cetera. So I think defining research questions is a challenging and interesting evolving concept, because of course it moves as you progress. But the approach I've taken is always think about a problem and the talent and the day in, day out passion you'd have for solving it, and then ask of others who are experts at solving that problem or who have that problem and want it answered if the way that you're approaching it or proposing to tackle it is as innovative as you think. And in my experience, I've found innumerable times that it's not that innovative. Someone's already tried it. Go to the literature, read a lot, and then revisit the original proposal. And you'll probably see, by being a student, remaining a student, and studying what's been done previously in the form of publications and books and talking with others, that you learn a lot in that process and your research really gets refined from there. Sorry if that was long-winded.
That was fantastic. No, what I want to share is that I was always admiring those people that had a purpose, like a research that they want to pursue. And I was always feeling anxious, and when going to a new lab or a new space, and not having my own ideas to work with. But no, you don't necessarily need to have them. You can just join a lab, see what they are working on, and then your ideas will come up. It's just curiosity. If you are curious enough and you like science, those questions are gonna come up. And of course, as Vali said, after reading a lot and seeing what is there, what is published, the questions come up. Mm-hmm.
Thank you both. You've already given many pieces of advice today. Our next question asks if you could give one piece of advice for your professional career path when you were young, what you wish you had known, what would that be?
I think what helped me most in communication and writing and in thinking was just reading a lot. And I picked that up more so as I was preparing for the MCAT, so that I could read through these questions and answer them and pass that hurdle. That, I mean, if I were to take the MCAT today, I'd probably get whooped. But that being said, the skill set I learned, I wish I told myself earlier, was that if you just absorb books and spend an hour or two a day dedicated to some topic, you'll get a lot better at it very quickly. So one thing I did in high school that I do to this day is I read The Economist every week, and I read about what's going on in the world. And I feel like it gives me perspectives and appreciation for the ways in which technology or fossil fuels or social crises or, you know, trade relationships between countries are different problems than the ones I'm focusing on today, and give me a context for, you know, where society's headed and how I fit in, and then also an appetite for when I go to the literature to read a publication end to end and learn from it. So quite simply, it was, if I could tell myself at a younger age, read a lot more, and have picked up the appetite for learning from what's written. People put time into thinking how they communicate, and you learn a lot, not just from that content, but from how they're communicating. And that's a key skill set, I think, to be able to communicate with others as you go, both in written and in verbal formats.
Wow, nice. I need you to give my young myself that advice.
I'm telling my 6-month-old that.
But I would also add that I would have liked to be more less scared and more flexible in my decisions. And you can be wrong in the first choice of your path, and that's okay. Or you not necessarily had to be wrong. You can choose a path and then use that learning for a new path, and that's okay as well. So the flexibility and the ability to adapt to your new yourself. Because you're going to change a lot.
Thank you both. I'll ask one last question, and we've asked in previous sessions before I close and hand it back to Olivia. Obviously, whether you go to medical school or graduate school, it's a long training path and one that can feel intimidating, isolating, frustrating, exhausting. So throughout that journey and today, how have you balanced your work with your life outside of work and tried to strike that balance?
Should I go first, Vali?
Yeah, I think go for it.
I think there are weeks and weeks, month and month. Sometimes it's easier than others. Sometimes there are a lot of work or a lot of things to study, and that's okay as well. Just to acknowledge yourself that it's going to be just a time where you have to put all your effort in, but then you can just use the extra time in other weeks to do what you like. What helped me a lot always was to have my friends around, my husband reminding me that I like to be outside, that I like to bike and just camp and that stuff.
Yeah, I think balance is key in any of these careers, to continue feeling like you have the energy and fresh perspective and enthusiasm for what you're doing. For me, I think building in purposefully an hour of exercise every day is one way in which I force balance. And as life has gone on and I got married and have a child, my priorities have shifted to make different allocations of time a priority. But when I think of in my training years and how I found balance, it really was forcing exercise and healthy eating as a must-have, and noticing very quickly when I didn't have that, how much the quality of studying or the energy to go out with friends on a Friday or Saturday after I'd earned it kind of eroded. So I think diet and exercise is certainly how I mandated balance when it got really busy.
Well, thank you again so much, Cecilia and Vali. This was wonderful. I'm going to hand it back to Olivia in case there's anything you'd like to say to wrap us up. But thank you all again for coming.
Thank you so very much. This was such a great presentation. Again, thank you, Pano, Cecilia, Vali. And I actually have a quick question for Vali. We actually did a field trip with some of our students to Illumina. I believe, well, it was right before the pandemic. And I think it was just such a great experience in such a, you know, very, very interesting company. They're doing a lot of really interesting things. I don't know if you're still with them, and you know, but I think your trajectory is just so interesting that you have the engineering, the private industry, and then you know, being a doctor. And I don't know if you know, my question is, have you found that you prefer one over another?
I think they can't work without each other. It's not like when we prescribe medicines, it didn't come from a pharma company, or when we use pipettes that someone didn't make the tips in a supply chain that necessitated getting it. And I realized academics and industry tend to view each other as in completely different worlds, but they're absolutely dependent on one another just as much as I'm dependent on nurses or real PhDs like Cecilia to guide the research, right? Like there's no way I'm going into the lab telling Cecilia how we do an experiment. I'm asking her, how do we progress here? So my answer to that, and with industry, is it's totally symbiotic. Without industry, I mean, you can probably look around your room, everything's made by someone down to your computer. So we definitely are relying on them, and it's a wonderful relationship that I think is healthy for society, and we're so fortunate to be in a country where that's celebrated.
Yes, no, thank you for that. And I think, you know, I often tell students, because sometimes they think, okay, I want to be a doctor and I cannot deviate from that. And so just hearing your stories and how you started and the different, you know, it's not a linear trajectory, and how those things changes is very helpful. So thank you so much for your presentation. I just want to remind everyone that we will be posting the recording on our website. So if you want to access it, you can go to our website, which we put in the chat, and just, you know, play any part or the whole presentation. So thank you again.
Thank you, Leah.
Thank you.